Hello folks and welcome to the plasma membrane. Okay, so before we get started, I do want to show you or talk about the little areas that says flashcard moments here. A flashcard moment just means, hey, this could be a slide that's very important. So you might want to get a flashcard out, write the title of the slide, and then on the back, write one sentence or even some kind of example or even a drawing in your own words. And the purpose of that is that if you only are listening to my voice from other folks on YouTube, or even the voices only from the textbook, you're not building your own voice for the information. And a lot of times we may get caught up thinking we have to write an answer a certain way, when in reality, we just want to know, how did you process that information in your own brain? Okay, folks. And when it comes to YouTube, don't forget, if you go to the home page and you click on note taking and study strategies, you will see, if you scroll down to the bottom of that page, that I did provide you with a bunch of different YouTube channels that you can use, just in case my voice or the other voices you're hearing are not really gelling with you. They're not connecting. So it really is your choice to be able to move forward and figure out how you want to grasp this information. Okay? Now, back to what we were talking about, the good old plasma membrane. We already know that the plasma membrane is going to be that permeable or selectively permeable barrier. So what does selectively permeable mean? It means only certain things can come in and only certain things can go out, because this membrane surrounding all that stuff inside the cell. The stuff inside the cell is going to be that cytoplasm, the cytosol, the organelles, the nucleus, all the stuff inside. And the plasma membrane is going to be surrounding that. You can also think of it as like, if you had a clear bag and you filled it with water and you put a bunch of like Legos inside of it, you should be able to see that the clear bag is going to be a barrier between what's inside and everything that's outside. The only difference is, when it comes to a plasma membrane, you would take a little bit of like maybe a pin needle and poke holes in that bag, and some water will come out and some stuff will stay in. And so you'll be able to see that when it comes to the plasma membrane, just to visualize it as well. Okay, folks, so just keep that in mind. Now, here is a picture of what it looks like. Okay, and of course you can get like microscope pictures, which are way better than sometimes the cartoon versions. You can see that they're made of three parts. They have lipids, they have carbohydrates, and they have a lot of proteins. And yes, these are those same macromolecules we talked about in chapter 2. So you want to make sure you're going back and getting details for those molecules if necessary. Okay, folks? And really, lipids are the big piece here, because lipids are the thing that helps with the overall structure. Carbohydrates and proteins come in, they have their own functions as well to help it be selectively permeable. Now let's go ahead and look at some of the details of where lipid is. So there's two types of lipids that we focus on here. Well, really three types. We're going to have our good old phospholipid. We also have cholesterol, which comes from those steroids, which are, of course, lipids as well. And then we also have glycolipids. So yes, we learned about the four macromolecules: lipids, carbs, and proteins, and of course, nucleic acids. But guess what? Some of them can combine. So the glycol part here means that you're dealing with something that's a carb, and the lipid part means you're dealing with something that is a lipid. And the glycolipids can come together where sometimes you'll have a phospholipid here and you'll have some kind of like carbohydrate coming off of it. And a lot of times we'll talk about how these glycolipids can help with recognition of other cells, or even a small amount of cell communication as we go along. Okay, folks, so this is what it looks like. This is what it's made of. We're going to be getting the anatomy of the plasma membrane as we go along, but I want to focus on what is the function overall of this plasma membrane. You can also call it cell membrane too, but it's easier when you talk about plasma membrane. It'll force your brain to be like, oh, plasma, what is it made of? Now what's the functions? Now what is this? Now what is that? So it is up to you. Another thing as far as details you'll see is, because we're talking about a cell and we have everything inside the cell, you have two types of fluids. You have all the fluid or the watery environment outside the cell, and then you have everything that is going to be inside the cell. And the plasma membrane is going to regulate what comes in and out, and that's what the whole goal is. Next, we're going to go through the four main functions of the plasma membrane. As you can see, we're going to start with number 1, then we'll go to 2, 3, and 4. So first, just like we said, it is a semi-permeable barrier. So it is a physical barrier that's going to enclose the inside of the cell from the outside of the cell. So again, we have our stained plasma membrane, and definitely we're going to talk about what all these things are, but let's get the functions first. And then we have things that are outside the cell and all the stuff that is inside the cell. As you can see, we say we have a watery environment inside and outside. Now, for some folks, you'll be like, I already know what a phospholipid is. I know that we're dealing with a polar head and two non-polar tails. I get that already. So you might already have information to kind of connect. But we're going to talk about that piece by piece, okay? So first things first, physical barrier. That's our first function. The next function is selective permeability. What does that mean? Because of its actual structure, What is it going to do? Dictate function. So the structure of the plasma membrane is going to be a bunch of different phospholipids in one layer and phospholipids in another layer. So how many layers of phospholipids do we have? We have two. We call it a bilayer. And these bilayers, one layer here and then one layer at the bottom, come together to help form the overall, structure of the plasma membrane. And then, remember, we're not just dealing with lipids at this point. We will have some proteins that can go across the overall bilayer or just be embedded into the bilayer. And again, we also have other lipids like cholesterol as well. Let me get another color here. We also have little cholesterol that can be, of course, within or embedded in these phospholipids as well. And so you'll see cholesterol here, kind of between these phospholipids. And we'll see a better picture of this as we go along. Here are some proteins here, other proteins here. So we'll be seeing more details of that as we go along. Okay, folks? And because of its structure, because of the proteins, and because of course, those glycolipids, meaning extra carbohydrates attached to some of the phospholipids, we have the ability to determine, hey, which substance is going to enter, which substance is going to exit. Let's get an example. Let's think of good old water. We talked about water in chapter 2. Okay, water is polar. The phospholipid has a glycerol head, which is also polar. So this is polar, this is polar. All these are polar in this bilayer. So if water can go ahead and reach the polar area, but when we start getting to the non-polar areas here, the non-polar regions, water's going to have a problem. So let me write a larger one up here. Here's the polar thing. Here's our water. Water wants to cross the membrane. Well, it can basically interact with the polar portion, but it will not be able to interact with the non-polar fatty acid chains that are part of these phospholipids. So then you might be asking yourself, though, what do we do with that? Well, if water can't enter using just basic diffusion and going across this bilayer, it's going to need some help. Water is actually going to use a transmembrane protein, which goes across the entire bilayer so it can get into the cell easily. And remember, water is very important for us. So we want water to have an ability to get into those cells. And you'll learn, it depends on the information that you want to kind of go into depth with. But in this chapter, you'll learn that those membrane proteins are called aquaporins. Aqua for water and porins because it's like a little pore. A pore that's in between these bilayers so water can easily be going in and out of the cell when it needs. Okay, folks? So that's what's really cool about that. But if you have something that is going to be non-polar, like say you have an extra fat or something like that, it has the ability to, of course, interact with the non-polar regions in the bilayer as well. And you see we have a bilayer. We have phospholipids here, and we have phospholipids here, and that's where that bilayer is coming from. So selective permeability is very, very important. But as you can see, it's going to take lipids, it's going to take proteins, and of course, it's also going to take carbohydrates to help us be able to determine those things. Let's move on to the next function. Our next function is good old communication. Now, communication is twofold. Communication is going to first have to happen with the actual proteins. And here's the beauty of it. Remember I was telling you about those little messengers that get inside the cell at the end of the other video? Well, guess what? Those messengers can also be hormones. And hormones need to be able to go into a cell and be able to communicate to send a message to certain parts of the cell. So here's my little happy hormone. And this hormone needs to be able to have certain little proteins on it that certain proteins in the plasma membrane can recognize. So let's go ahead and take this particular protein, and let's say it has a little area for our happy little hormone. Our happy hormone is going to interact with that protein, and then guess what that protein can do, folks? It can change shape. It can change shape and take that message and send it inside of the actual cell, which is insane, but it actually happens, which is pretty cool. And sometimes you can even think about this going back to that term I was telling you before, signal transduction, if you're really into biochem and stuff like that. That's my background, which is biochem. So of course I get a little bit too about it. But just know that it's really cool to know that, hey, there's a way that the cells are communicating, even at this small molecular level. And so this happy little hormone can go, this particular protein can change its shape and create a little messenger that can go to wherever part of the cell it needs to go and send its message. That is very cool. Okay? So just keep that in mind. There's also a way for cells to recognize each other by these little glycoproteins that are attached to some of the phospholipids. We were talking about how you can have a carbohydrate attached to a phospholipid on the membrane. Well, yes, they also can recognize different cells. And sometimes they need to quickly speak with one another and say, okay, you may need this, let's talk about it. And that's just really cool about that. So it's not really going to be 100% just the proteins. They're going to do a lot of the majority of the work. But of course, you do have those little glycolipids that can help with the recognition of the cell. Okay. And let's just give an example of that. So if you have a skin cell, you want it to be able to interact with other skin cells if you know you have a cut on your arm from them, right? You don't want like a cell from some other organ system to come up and be like, hey, now we're including ourselves here. That wouldn't make sense. That's why the connection and communication and recognition of cells is very important. Okay, folks? Let me stop talking. Let's go ahead and get into more detail of cell recognition. And yes, again, they do call out the glycolipids. And I just want to say you also have glycoproteins, because cell recognition is not just about just having the carbohydrates attached to the phospholipids. They also can be attached to proteins. And that, again, if we know proteins are all about communication and helping with communication and sending messages inside and out the cell, well, guess what? We can have glycoproteins that have the little carbs there that can help with cell recognition and communication, which is also really cool. Okay? Now, anytime you see carbohydrates, whether they're coming from a protein or, of course, we normally see it on a phospholipid as well, we call it the glycocalyx. And that just means that you have a bunch of carbohydrates attached to the plasma membrane to help with that cell recognition, which is also really cool. Okay, folks? And again, cells need to be able to recognize each other. If they can recognize each other and say that there's two skin cells that are next to one another, then guess what? These proteins can then come together to form tight junctions, tight junctions to create tissues, folks. That's why all this stuff matters, because we're building up to our hierarchy. We starting off with our chapter 2 stuff. Now we're getting to chapter 3. Chapter 4 is going to be tissues. Chapter 5 is automatically organ system, integumentary, our skin. So we have a lot of little details we have to lead up to that's going to help our brains kind of layer the information to build on these details. Now let's get into the anatomy. You see me drawing little stuff here and there. Let's go ahead and get into what these phospholipids are, what some of these proteins are, and get into those details. All right, the anatomy of the plasma membrane, meaning what is this whole thing made of? We know the functions already. We have some details on that. Now let's get into the details of what it's made of. First things first, you can categorize them into 3 different macromolecules: lipids, proteins, and carbohydrates. They all are going to have their own function and, of course, their own structures as well. So first, let's focus on lipids, because we know that's what the main thing. If you were learning about phospholipids in chapter 2, you know the main example was like the plasma membrane. This is your chance to make those connections. So first, we know we have a polar head. Usually that's going to be our glycerol. Why am I always talking about glycerol? Because we're talking about a regular fat. You have glycerol with 3 fatty acid chains, but a phospholipid is a modified version of that. It's a glycerol with 2 fatty acid chains. And these fatty acid chains are nonpolar. And of course, when you think nonpolar, you want to be thinking hydrophobic. So that is a fear of water. That's where the hydro comes from. Versus glycerol, which is going to be hydrophilic. Philic, coming from the Greek terminology, it means loving or loving toward. And so it really likes water, has no problem with water. That's how we get a polar head and 2 fatty acid nonpolar heads. Okay, folks? Now think about it. If one side of you likes water and one side of you don't, what do you need to do to make sure that the non-polar side does not get involved with water at all? You form a bilayer, okay? That's the reason why the bilayer is formed this way. Write a couple of them here, I'm just drawing out. And draw the best you can. I'm giving stick figure, and guess what? I'm okay with that. So you draw whatever way you want. For the folks who are really great artists, go ahead and just do what you got to do. That's cool, right? So just keep all that stuff in mind. Now I'm just drawing a couple of them here and here. But if I draw it this way, then guess what? Everything that's non-polar is connected. And then everything that's on the outside that's polar has the ability to interact with water. Why does that matter for cells? Well, remember, we said that we have the plasma membrane is basically a barrier for everything that's inside the cell. We have a watery environment inside, and of course we have a watery environment outside. We have fluids, blood, etc. And now we have the ability to interact with the watery environments inside with all of our polar heads forming this way, and also the watery environments on the outside with all of our polar heads facing the outside. Now, of course, you can call this intracellular fluid, that's the watery environment on the inside, or extracellular fluid, which is the watery environment on the outside. Now, sometimes they create the extracellular fluid. And they call it the extracellular matrix because not only does it include fluid, it includes those glycoproteins and it includes other attachments to help the structure of the cell be formed as well. And so we'll get into some details about the cytoskeleton and those structures as we go on. Now, of course, you can also add glycolipids in the lipid section. And we know that's all about cell recognition, okay? And also you can add glycoproteins here as well. That's going to help us with the cell recognition and, of course, communication, depending on what we're doing. Let's talk about one of the other biggest pieces of the plasma membrane when it comes to lipids, and that is cholesterol. Cholesterol is very important, so let me go ahead and erase all this so we can get to chatting. Now, cholesterol is one of the most important portions of the actual plasma membrane because let's think of some different environment. If we had our plasma membrane, which is made of fats or lipids, right? Phospholipids specifically. If you have the ability to increase the temperature in any way, like if you're running, your body temperature is going to increase. Do you want your cells to just fall apart and become way too fluid? Meaning that the phospholipids are next to one another now has the ability, if it's too hot, to be further away from one another. If they're further away, then the membrane is not going to be as selectively permeable and stuff just kind of goes through. You don't want that. You don't want just anything going in your cells when we have this highly regulated system, right? So what happens is we have some cholesterol. And the cholesterol is there to help, again, stiffen that membrane, but really is there to make sure you control whether or not this membrane is going to be too fluid or too rigid due to changes in temperature. And we're human beings. We're changing temperatures constantly. And our environment, of course, influences our temperature as well. So the cholesterol is going to be like, okay, you guys can do your thing. Like you can move around because phospholipids in the plasma membrane have the ability to move around. They can even switch places. They can go from the top layer to the bottom layer, which is also awesome. So cholesterol is like, you can move around a little bit, little tiny little bit, but you ain't going nowhere that much. Okay. So we're going to help stiffen that membrane so it doesn't have those issues. That's if you're increasing temperatures. You can do the same thing when you are decreasing temperatures. Ooh, I'm really, really, really cold, right? So when you're decreasing temperatures, you see the same thing, except the opposite, right? So let me go ahead and I can do my erasing. I'll get this here. I'm going to decrease the temperature now. Let's see what happens. When I decrease the temperature, now my overall phospholipids are too close to one another, almost smash-like, and become super rigid. So if it's too rigid, guess what? Selective permeability, one of the main functions of the plasma membrane, it's out. So you don't want those situations, right? So what do? We have our good old cholesterol. Cholesterol is going to come through and say, yeah, I know you're cold, but you ain't going to get that close to one another. And that's the beauty of that cholesterol. Okay, folks? The reason why I'm speaking to you in this way is because remember, you want to have your own voice for this material. That's how you remember things. Another way to remember things is just thinking of, health issues that can really relate to, hey, I don't have this. Like, that's what we'll get into when we get into the organelle. Hey, if my mitochondria is off, what kind of issues can I come into? Da, 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 da. So that's just another way to just remember those things. And that really is going to be your lipids. Now, let's think about it. Why else would cholesterol be good? Of course, it's different than membrane, but one thing is cool if you're thinking of like blood. Blood has the ability, of course, to move through your arteries and your veins, but it also has to go through capillaries. And the capillaries are very tiny. It only allows one red blood cell at a time to go through. So the cholesterol helps to squeeze and make the changes in the actual plasma membrane of the red blood cells so that they can squeeze through without breaking apart. And that's something that's awesome as well. And then once they get where they need to go, maybe they're going from arterial blood to, of course, venous blood, then, of course, they can go back to its original shape. And you might be saying, well, how is this all going to work? Well, guess what? The plasma membrane, we also call it a fluid mosaic model, which we're going to get into next, because all these pieces that we're seeing here have the ability to move. And their ability to move helps, of course, dictate their overall function. That's selective permeability, the ability to communicate, of course, creating that physical barrier, and, of course, recognizing the cell. Now, let's go ahead and get into the protein of it all. Just like you learned in Chapter 2, proteins are the most ubiquitous substance in our body, or most ubiquitous macromolecule in our body as well. Okay, folks? That means it has so many different structures, which therefore has so many different functions, and that's no different than the plasma membrane. The plasma membrane has proteins that have so many functions. I'm going to tell you as many as I can, right? So for example, transport, communication, joining each cell together. Of course, that's going to be something that we talked about as well. Let's get into as many as we can. So transport. Proteins can transport. Now, what does that mean? It means it has the ability to allow certain substances to enter or exit the cell. That's the main thing, okay? It also can act as receptors for chemical messengers like good old hormones. Remember I was telling you about signal transduction. We'll bring that in again. If a message from the outside needs to get into the cell, that message, that happy hormone, will go around to the protein that has a receptor that can recognize it, and then that message can then go through and cascade down into the cell where it needs to be. So that's another beauty of proteins as well, as receptors. Okay, let's think of other things. Well, we already know one of the big types of proteins we focus on are enzymes. So yes, some of the proteins that are embedded in the plasma membrane has enzymatic activity. So what are enzymes? These are huge globular proteins that have these active sites, just like we saw with some of the receptors that certain things can go into to speed up chemical reactions. So we know it's not a receptor specifically, it's a specific enzyme embedded into the plasma membrane. Guess what it can do, folks? It can take different reactants and form specific products faster than if you had a reaction on its own. And that's the whole goal of enzymes. I know you went through those details for chapter 2. What else can the protein do? We already talked about cell-cell recognition. We know we can have glycoproteins that help with cell-cell recognition, and really just identity. Again, it's very highly regulated. Okay, only certain people are getting in. And so what that means is like we're definitely dealing with the fact that yes, if you don't have the certain type of receptor, or you don't have a certain messenger attached to you, or you don't have the same type of structure that I'm dealing with as far as my cells over here, you're not going to be able to send any message to me. That's the way it's kind of working, which is really cool. We can also say that it's a part of cell-cell joining. Yep, there's proteins that help create cell junctions, which we're going to get into a little later. And those cell junctions are going to help us create tissues. So it actually helps join these different cells together to form those tissues. So proteins in the plasma membrane can do that as well. And another thing that you see here about the extracellular matrix, well, proteins can attach to the cytoplasm inside the cell. So we're not the cytoplasm, I'm tripping. Cytoskeleton. Okay, so there's like, we have our plasma membrane, which is great. And then there's a bunch of protein fibers inside the cell that provides its structure. Remember I was telling you about that clear bag, and imagine that you put water in it and a bunch of Legos? Well, now we need to make sure that the bag is not just squishy all around. We need to make sure it has a structure so that I just can't squeeze it and do whatever I want. I have a specific amount of proteins in there. So imagine you take like a bunch of, you break pieces of spaghetti and put it into that bag. Well, that's going to make it different. It's not going to make it be able to do it any kind of shape you want. It's going to make it have more structure. And guess what? The proteins in the plasma membrane have the ability to connect to some of that cytoskeleton so that the structure is upheld. Okay? So you might be thinking, oh, girl, there's too many protein functions. I don't have time for this. Well, don't try to remember every single one of them. Do your best to at least remember 2. I would just focus on enzymes because I already talked about that in chapter 2, and I just want to layer information. And maybe I'll focus on receptors or transport. That's one thing that can help you kind of get started. And as you go along, maybe you want to keep layering on different functions, but you don't have to do everything all at once. So that's why I want to just make you feel comfortable with saying, hey, do your best to at least memorize something to put it all in your head and then move forward that way. All right? And of course, folks, the last thing we want to focus on is carbohydrates. And again, the way we have carbohydrates when it comes to our plasma membrane is that they're usually connected to some of the lipids or proteins that are already part of the plasma membrane. And they're usually going to be on that outer surface only. So they're going to be the tops of these proteins, or they're going to be at the top of this particular phospholipid. Not going to be usually on the inside. Okay, why? Because they're all about recognizing the cell. Remember we said, who are you? You cannot come in if you're not a cell that's similar to me, because you don't want tissues being formed from a muscle cell, a skin cell, and then a blood cell all coming together. You want to make sure that actual functions that are happening. You know, if I make tissues for muscle, make tissues for skin, yes, we need to make sure we can actually identify the difference between the two. Okay, folks? Another thing that's really important is that these carbohydrates or glycoproteins, or the glycocalyx that we see here, like that coating, helps us basically understand which cells are there to aid in the process of keeping homeostasis, or which cells are there to basically ruin our life. What that means is they need to be able to recognize if they have pathogens, which can cause sickness for the cells and kill cells as well. So if they can recognize pathogens, they can say, hey, we need an immune response over here. And that can be something that's very important. Now, table 3.4 is very important. And this is the table that I'm going to be going through as we continue to go along, because this table is so great in putting the structure and function of the plasma membrane, the nucleus, all the organelles, the cytoskeleton, all those details into one little area. And personally, if I was doing, back in school in this chapter, I would go straight to table 3.4 and then add in notes for myself or add in things I can remember from it straight from that table. And everybody can do their notes differently, but that's just how I would approach it because there's so many different variations. And yes, you can go through the whole entire textbook and be like, mitochondria does this, Golgi apparatus does this, smooth endoplasmic reticulum does this. And then you might be down 3 hours and still be like, what does mitochondria do? Versus you have one sheet of just this table that you print out, and then you're adding notes to it, and then you're continuing to reorganize or revise those notes, which can be helpful. But if you go to table 3.4, you'll see it has the plasma membrane here. It's telling you it's made of a double layer of phospholipids that have cholesterol and proteins embedded, and the proteins can be embedded across the entire membrane, and of course just embedded in the membrane itself. And of course we also have carbohydrates as well. And then it gives you some of the functions that we already talked about. You can either make the functions more simplified, like we said before, you can say physical barrier, selective permeability, communication, cell recognition, versus having to write the whole sentence down. So that is really up to you. Now we'll go ahead and get into the fluid mosaic model, which basically helps us understand, or it's kind of a model or theory that is used from scientists to say, hey, this is what the plasma membrane is about. And then we need to be able to at least draw it out as well. So let's get to that next. All right, folks, so fluid mosaic model of the plasma membrane. It's just telling us, if you really want to say, what is the plasma membrane all about? You can literally say that it is a phospholipid bilayer with embedded proteins arranged as a fluid mosaic model. You can simplify that. You can even add in glycolipids and all those things as well. Let's break down what fluid is. Fluid means things can move. Mosaic means it has many parts. So everything you see here, the phospholipid, the proteins, whether it's a transmembrane protein or integral proteins that are like embedded in there, and also things that have like this glycoprotein here that has a carbohydrate attached to it, all these things have the ability to move. Yes, this phospholipid here can move here. This phospholipid down here can move up here, and they can move around. So you might be thinking, how do we actually keep this membrane together when they all can just do whatever they want? That's the beauty of it. Okay, it's flexible, has many parts that can move around, and guess what? It's all there still to create the barriers we need. But it also helps us understand what that selective permeability is all about. Certain things can come in, certain things don't. Okay? And so it's something that you really want to know. And also you really want to know like how to do a quick drawing of it. Again, you don't have to be, the best at it. You just want to have it in your head. You want to be able to say, okay, I have some, let's just write them down here. Some phospholipids. I'm just going to write a couple of them. I ain't going to go all the way. Okay? Some phospholipids. Boom, boom, boom. I'm going to add some cholesterol in here. There we go. I'm going to add a transmembrane protein. There we go. And maybe I'll add an integral protein as well. Okay? And then I might just go ahead and add me a couple of glycolipids and maybe a glycoprotein here as well. Okay, folks, and how do we make it a glycolipid or glycoprotein? You add a carbohydrate on it. Okay, folks, so you want to be able to draw the plasma membrane. I would say make sure you know how to focus on the different parts of the plasma membrane. Excuse me, not parts, functions of the plasma membrane. And then you want to be able to draw it and be able to identify different pieces. Like you should know that this is phospholipids here in the bilayer. This is the protein. This is the glycoprotein because it has the carbohydrate. This is the cholesterol, et cetera, et cetera. So that's really how my brain would organize this stuff. Okay, folks? Let's move on to our last piece when it has to do with plasma membrane, which are cell junctions. All right, folks, so here's the last portion of this plasma membrane video. As you can see, we still have another flashcard moment. Sometimes I'll remember to say it. Most of the time I probably won't because I'm out here chatting. All right, so cell junctions, why are they so important? Because remember, we can learn all about the cell and the plasma membrane. We need to know how cells can come together to form tissues. How do they do that? Through cell junctions. So it's almost like you're taking a cell and connecting it to another cell after, connecting to another cell, another cell, another cell, another cell. And guess what? You can form tissues like muscle, skin, et cetera, et cetera. And they stick together. And if they're sticking together, guess what? They can communicate. So you can have stuff going in between this cell, going into this one. They can communicate with one another, maybe sharing different messages across one another. They can all communicate, which is pretty amazing, right? Just keep that in mind. Now, there's 3 different types of cell junctions we focus on, and there's also microvilli, which I wanted to bring into this plasma membrane discussion because it is an extension of the plasma membrane. Well, first, let's talk about the types of cell junctions. First things first, we have tight junctions. And what I like to do when I see this information is I need to just find one word to help me understand what these different things are, okay? You'll see that when I talk about organelles. Like, if I see organelle, what is it about? Just automatic. Sometimes that helps, and then I layer in other notes as I go along. So tight junctions are going to seal cells between one another. They're going to create a seal between 2 cells. Desmosomes are going to create an anchor between cells, and then we're also going to have gap junctions which help with communication between cells. Okay? So when I was writing all these little lines here, if I want to have communication between cells, these will be our little gap junctions here. If I want to seal the cells when they're next to one another, that will be our tight junctions. And of course, I want to anchor the cell, maybe on this matrix or something like that, or anchor the cell between the 2, just to provide more stability, then I would definitely have my desmosomes there. And all of these junctions are important, of course, to be able to create cohesive tissues. So let's focus on the tight junctions, which are all about sealing. As you can see, there's going to be proteins that create a seal, and I'm going to just circle the seals here. Let me get a better color. Circle the seals, and you can see if something is sealed, that means it's not going to be selectively permeable. It's going to be impermeable, meaning things are not going to be able to get through, okay? So it has different seals that you can see because of different proteins that are allowing it. And then, of course, there's going to be space between the cells in some kind of way, but the tight junctions that are created are going to help seal that, okay? So let's think of something that can help us remember tight junctions. Outside of this, the first thing that comes to mind is going to be the intestinal lining. So when you have your intestinal cells, you know they need to be able to have the ability to stick together in that way or seal to one another. And that's one way you can have tight junctions as well. Another thing we can think of is the blood-brain barrier. Yes, you want to make sure that it's a seal, but you also want to make sure there's space to move things in between it. Same thing with the intestines. It's sealed, but you also can move things in between it with those spaces. Okay? And we can even go further and say, okay, well, what if we actually have some kind of disease that is going to break some of these seals? Well, let's think of in the intestines. If you start breaking some of these seals, that means you're going to have more space, right? More space compartments. More space compartments can mean that you're actually moving fluids or nutrients, for example, aren't going to be absorbed as much. Maybe they're moving in and out. Think of something like leaky gut. And that's one thing that can happen because you're dealing with issues with your tight junctions. All right, folks, let's move on to desmosomes. Now, desmosomes, we said, are going to anchor. They're going to help anchor the cells when they are connected to one another. All right, folks? And that means they're going to bind cells together. It's almost like the perfect example that I see here is going to be like Velcro. It binds it together. We know with Velcro, it's going to take enough force to pull the actual Velcro apart. So that means you're definitely creating some kind of tightness when it comes to this anchoring. Okay? And again, it helps cells from tearing apart. Now, where will you see a lot of desmosomes? We're thinking the skin, the heart muscle, for example, you'll see some desmosomes. And just talking about just talking about the skin itself. Let's say you have issues with desmosomes. Then guess what's going to happen? Like, say you have some kind of autoimmune issue that attacks your desmosomes. Then that's going to cause a lot of blistering of the skin. Because again, you do not have that Velcro there. So you can also create like more interstitial fluid here and cause blistering. All right, folks, let's go to our last one, which is going to be gap junctions, the communication channels between our actual cells. So our gap junctions are going to allow us to have messages go in and out of the cell itself, which is great. Now, with gap junctions, I always say, well, we know it's all about communication. What are some areas where we need to make sure we're communicating well? Think cardiac muscle. Cardiac muscle, the heart, you want to make sure you're getting all of your communication between the heart cells so the heart can do its job. Also, smooth muscle as well. Because we have things that are happening on auto, for example, like of course your heart is pumping on its own, you don't need to actually add anything to do the pumping, or what smooth muscle like in your bladder is happening on its own, you want to make sure the communication is really good. So let's kind of think of some ideas behind where things can go wrong in the cardiac muscle. So if you have issues with your gap junctions, and it's all about communicating and also allowing ions between the cells, and those ions are going to be important for creating those potentials or cell potentials inside the heart itself. And guess what? If you have issues with gap junctions and you can't get those ions and communication to happen, that's where you see a lot of folks have heart arrhythmias. So it's little details like that. Yes, you can talk about gap junctions being communication, give a little bit of an example, and then go ahead and go and get a health exam. That can help your brain kind of connect the information as well. Now, the last thing I want to focus on is the microvilli. Now, microvilli are these little tiny finger-like extensions of the plasma membrane. Not every cell has them, but of course, if we're talking about the intestinal lining, then yes, we do have microvilli. Because the main function of microvilli is to increase the surface area for absorption. That means there's more areas to absorb nutrients. And when you have something like food digestion, which happens in the small intestines, you want to make sure you're getting as many nutrients absorbed as possible, folks. That is the goal. Now, these microvilli, again, are extensions of the plasma membrane, and there's other extensions of the plasma membrane we'll see as well, like cilia, which helps move cells, and also flagella in the sperm, which also helps move the sperm cells. Right now, we're going to focus on the microvilli. And these microvilli, again, can have their own, glycocalyx, meaning that they have their own little carbohydrates attached to them as well to help with recognition and getting certain nutrients across, which is pretty cool. And one good example I can think of when it comes to microvilli is from the intestines itself. So think about it. If the intestines have microvilli to make sure there's more of a surface area to absorb nutrients, then let's think of a disease that damages or doesn't allow for a lot of absorption. Another thing you can call it is malabsorption. And one disease that causes malabsorption in humans is celiac disease. So you're not going to be able to absorb all the nutrients coming from the food you eat, all because the microvilli are damaged in some way. Okay, folks, so I'm going to end this video here because, of course, I'm back to chatting. But the plasma membrane is just very exciting. It's very cool to learn. Just try your best to make sure you organize the information and make it quick for yourself. I have a lot of flashcard moments, but I want you to know that you can even take the plasma membrane and talk about its function on one flashcard, take the plasma membrane and talk about its structure and label it, and then put details about lipids, proteins, and carbohydrates. It really is up to you, folks. But I know you can definitely get through this, and let's go ahead and get to our next video.